Good morning, Honors Bio people. Today we're going to talk about the mutations in Section 4. We're going to skip Section 3. We're not going to talk about how genes actually function. The majority of Section 3 on gene functioning has to do with the lac operon, which is how a prokaryotic cell actually functions. And eukaryotic cells function a lot differently than that, are a lot more complicated, and quite frankly, I don't think it's something that we absolutely need to go through at this time. I'd like to get through human genetics before we finish up for the year for sure, and then get into a little bit of the evolution. So we're going to skip Section 3. I don't think it's real necessary for what we're trying to accomplish here. What we do need to talk about, though, is mutation. One of the things that we know about DNA is that it is controlled by a sequence of nucleotides. The messages on the genes that are located on chromosomes are dictated by what the sequence of the messages, what the sequence of those nucleotides are. Each nucleotide fits in a sequence of 3 nucleotides that make basically a DNA word, which we call a triplet. Those triplets are in turn complementary to a messenger RNA codon. That messenger RNA codon then is complementary to a transfer RNA anticodon, which codes for ultimately the placing of an amino acid in that position. So everything has to go right in order for that amino acid in that particular protein to be placed in the correct place. Sometimes mistakes occur, and as we get older, these mistakes happen more often because we don't correct our mistakes. We don't correct our mistakes as easily as we did when we were younger, which is why more older folks have more cancers. Don't correct our mistakes in replication and things like that as well as we used to. So what are these mistakes? Mutations are those mistakes. Those are changes in the genetic material. Those are changes in the code, changes in the sequence that you have the nucleotides. And there are several different ways in which we can get these changes. So mutations that occur produce changes, obviously, in what is ultimately produced, the kind of protein that is produced. Whenever this happens in a single gene, these are known as gene mutations. Whenever this happens in a large amount, in other words, we have large chunks of the chromosome being changed, we call those chromosomal mutations. Now, gene mutations come in a number of varieties. Those that are involving a change in one or a few nucleotides are known as point mutations because they occur only at a single point in the DNA sequence. So they happen at a very specific position on the particular gene sequence. Point mutations include substitutions, insertions, and deletions. Now, because they're only happening at a particular point in the gene, it might sound like, well, that's not going to be that bad. The problem is when you add things to the sequence, or you insert something that's not supposed to be there, or you take something out, you change the message. Kind of like having the word cat and taking out the letter A, you have CT, which means something completely different, which in genetic language means you're going to have a complete different sequence then, and you're going to have a completely different protein produced. Substitutions usually affect no more than a single amino acid. Why is that? Because whenever you get the triplets, and you take one particular nucleotide and you substitute it with another. In this particular case here, we took a cytosine and we substituted it with a guanine. Well, instead of having a particular amino acid, arginine, then you're going to get histidine. And while that may produce an abnormal protein, it's not going to be a huge lock-stock change. Okay, substitutions are only going to affect that one single amino acid. That doesn't mean it's not going to cause a problem. In some cases, very little problem. Some cases, it could be the same. A change could actually have the same amino acid put in that place. But overall, it's going to be a very small change. So it's only going to be one amino acid that's going to be changed. The effects of insertions and deletions are going to be absolutely more dramatic, and the reason for that is it's going to change more than just one triplet of information. Once you add something to a sequence or you delete something from a sequence, you're going to change everything from that point on, and that means every amino acid could be changed. So the effects are going to be very dramatic. The addition or deletion of a nucleotide then causes a shift in the grouping of the codon. So instead of having a particular sequence in a triplet, you're going to shift that and you're going to change every one of the triplets from that point on. And whenever you do this, you're shifting the sequence. When you shift the sequence, that's called a frame shift mutation. Frame shift mutations cause a change in every amino acid that follows from that point, unless that is in case that particular amino acid has the same triplet that ends up being substituted. Frame shift mutations can alter a protein so much that it is unable to perform its normal functions. In other words, that particular, let's say the protein it produces an enzyme that catalyzes a particular reaction, which causes another molecule to be made. Well, that molecule is not going to be made the same way because that or won't be made at all. Let's say, for instance, it's an enzyme that helps to catalyze or break down a substance that's in the cell. Maybe it's a toxic substance to the cell. Well, if that enzyme is not working, it's not breaking down that toxic substance, and then that toxic substance will then build up inside the cell, and it will cause damage to the cell. So the protein that's going to be produced is not going to be the same as it was before. So as you can see here, in the particular DNA sequence, if you insert in this particular DNA sequence right here, if you insert after the adenine in the first triplet, you insert a T, then you move the C over to the next triplet, and you completely change every triplet from that point on. So instead of having methionine, you now have a different amino acid here. Instead of arginine, you've got another one different. Instead of tyrosine, you have a different amino acid there, and then amazingly enough, you still get the same amino acid, even though it's been changed, because some amino acids can have as many as 2 or even 3 triplets, which code for the same amino acid. So, completely changes all the amino acids in the sequence from that point on. So, in the example here, in a deletion, the loss of a single base is deleted and the reading frame is shifted. So if you had, and we've used our English language in triplets to make the analogy here, the fat cat ate the rat. Well, in this particular sequence, we're going to take the H out, okay, and then we're going to shift over the nucleotide from the next triplet to make up that complete triplet. So we've completely changed down here, we've completely changed what those words are, and obviously it doesn't make any sense at all. And whenever we produce a protein like this, the protein doesn't make much sense in biological terms either. It doesn't function. Now, when we have large chunks of chromosome that are changed, we call these chromosomal mutations, and they involve changes in the number of the chromosomes or the single chromosomes themselves. Chromosomal mutations include deletions, duplications, inversions, and translocations. So it's going to involve the breakage of a chromosome and the shifting of it, or the addition of a piece of chromosome from another. So it's going to be large pieces of the chromosome that are going to be affected. In a deletion, this would involve the loss of all or part of a chromosome. For instance, if the original chromosome had genes A, B, C, and then D, E, F in that order, and we deleted B, we would then only have A and C over on the left-hand side. Obviously, missing a complete gene. You miss a complete gene, you miss a complete protein that's going to be produced, and it's obviously going to cause problems. In a duplication, you have basically a gene that's going to be duplicated, so they're going to have two of those. Now, in a lot of instances, we think, okay, well, if you've got one, you'll be fine, and if you have an extra one, then you'll be doubly good. Well, that's not the case here. When you double up, that can cause problems. So in this particular case, the ABC sequence here is ABBC, adding another gene to that sequence. When we invert, obviously we're going to reverse the direction of the parts of chromosomes. So if in the original chromosome we have A through F, and we change, or we invert or reverse the direction of a portion of that chromosome, we now have AEDCBF. Inversions can also cause problems, even though you have the same genes, they are not in the correct position, and because of that, that can cause a problem in what's being produced. One of the things we've already talked about in a past discussion is the idea of a translocation. Translocation basically is when one chromosome part breaks off and then it'll attach to another. A translocation happens in very common areas, and we don't know for sure why this happens regularly on certain parts of a chromosome, other than the fact that we know those areas are weak and they are prone to, during meiosis, they are prone to break off and reattach, and they like to reattach into specific areas. So in this particular chromosome, we have A, B, C, D, E, F, and we have a translocation where we have now shifted the gene sequence by an addition from a shifting the gene sequence around. We now have GHIDF. Now, let's go back here. One of the typical things that happens, the most common of translocations in humans, is called a 15-21 translocation. And basically what that means is that chromosome 15 likes to break off on the bottom of the chromosome, and it likes to attach to the bottom of chromosome number 21. Once again, we've still got have research going on about this as to why it likes to do that. Obviously, there's a weakness in the gene, in the chromosome area where it breaks off, but why it likes to reattach onto 21, we're not actually certain. The ironic part of 15-21 translocation is that it causes a disease called Down syndrome, and many people know what Down syndrome is. It causes a person to have some very characteristic physical features, as well as being mentally challenged. Ironically, the 15-21 translocation can also manifest itself in exactly the same way if a person has an extra number 21 chromosome. If you have an extra number 21 chromosome, we call that a trisomy 21. Trisomy 21 and translocation 15-21 actually cause the same symptoms. Once again, kind of a mystery, but It does. So translocations happen in very common areas, but they can happen just about anywhere. Just that they happen more often in some than others. Many mutations are going to have little or no effect on gene expression. It's like sometimes it can actually cause the production of the exact same amino acid in that particular sequence. In some instances, it can change the entire message and make the gene inoperable. Okay, the protein produced is not worth anything. It doesn't do anything good for the person. It can cause a major disease. Some mutations are the cause of genetic disorders. We know that very specific genetic disorders can now be tested for because of the fact that they are truly genetic changes that have caused the disease. And as time goes on, we'll probably have availability of testing for just about every genetic disease. This idea of evolution, or slow change over long periods of time in living things, cannot occur unless there are changes. And these changes occur on a random basis, and they occur all the time. So how do we change as living things over time? How do we adapt to changing environmental conditions? Well, we do that through the change that comes about through mutation. So beneficial mutations can produce proteins with new or altered activities that can be useful. And whenever this occurs and the individual survives and it makes them their survival value a lot better, then it's going to be added to that genetic content and it will be passed on to offspring because it's an improvement in their ability to survive. Something we haven't talked about, polyploidy, the condition in which an organism has an extra set of chromosomes. Let me go back here. Polyploidy can cause, in most instances, polyploidy can cause lots of dramatic changes, as you would expect in an individual. Extra sets of chromosomes, or an extra chromosome, is, like I said before, it doesn't, just because you have one chromosome of a kind, doesn't mean that it's a benefit that you have two of them, or three of them, or even four. A good example of extra chromosomes might be in a sex-linked situation, might be the situation where you have an extra X chromosome. XY is something that's called Klinefelter's male, and it causes very definite physical changes in the male. There's also a type of male situation where we have an XYY, where a person has an extra Y chromosome. Once again, it causes differences, but in this case, they're very subtle. So the subtlety or the major change that it occurs just depends on which chromosome it is, and that particular chromosome will dictate whether it's going to be dramatic changes or very small changes. The Y chromosome is a very small chromosome carrying literally no genes for normal development of a human. They basically carry maleness. So the fact that a person has an extra Y chromosome doesn't really change a whole lot physically about that person. But an X chromosome carries normal genes that all people have. So if you have an extra X chromosome, it's going to cause many more changes. Okay, so that's basically our discussion on mutations. Mutations, like I said, are part of the evolutionary process. When these changes come about and are thrust into a new offspring, those changes either allow that individual to survive better, or they have no effect, or they will be detrimental to their survival, and that will dictate whether that gene will be kept into the gene pool and passed on to offspring in the future generations, which is something we're going to be talking about when we get into our chapter on evolution. So that's mutations for today, and adios, amoebas.